and please mute your microphone while in the meeting during the presentation. If you want to ask any question, please type it in the chat box or wait for the end of the presentation so that we can collect all the questions. And also there will be time for a moderated discussion at the end of all the presentations. What else should I say for the announcement? If you have any technical problem, please contact Rocco Pignata, either in private message in the chat or writing an email to rocco.pignata@eatg.org. I think that's it. So maybe we can start. You can see the agenda of this meeting, and so we will start with a presentation by Jean-Daniel Lelièvre about the trial design, and also he kindly included some slides on the implications for MPA vaccination, which is the one used in this trial. In the context of the monkeypox infection, which is happening in Europe very fast as far as we know. So Jean-Daniel, if you want to start your presentation, please.
Yes, thanks a lot. So thank you for the kind invitation, and it's always a pleasure to be able to discuss with the community our clinical trial. So some words on this clinical trial, which is which is a phase 2 randomized placebo-controlled trials on the use of vedolizumab with or without therapeutic HIV/MVA vaccine in HIV-infected individuals who start their treatment either during primary or chronic infection. So as mentioned by Giulio, the trial has already begun at least in one country, in Switzerland, in the center of Professor Fabio Condotti, and expected to move on both in France but also in UK and in Germany. So what are the hypotheses behind the use of these two compounds? The first one is that the therapeutic vaccination will induce either de novo immune response, but also increase the previous existing response to improve the control of HIV replication. And we will combine this vaccine with vedolizumab, which is expected to have an impact on the viral dynamics by limiting the access of infectious virus to target CD4 T cell in the gut, and the gut is the site with the most available CD4 T cell to date. So the use of vedolizumab was firstly shown in the NHP model, and in one publication in Science a few years ago, showing that you could have a sustained biological control in those animals after antiretroviral treatment combined with alpha-4 beta-7 antibody therapy. However, 3 other studies conducted in macaques did not find the same kind of results. However, while those studies were not conclusive in macaques, other studies have been performed between the two, between the first NHP study and the third studies that have been performed after the first one. 2 clinical trials were performed in human. The first one was performed by the NIH. You can see the main entry criteria in this clinical trial that was to be infected, obviously, by HIV, stable on CL for at least 2 years, with CD4 more than 450 cells per millimeter cube. The restart criteria, the schedule, we will see the schedule of this clinical trial, and it's associated with the infusion of vedolizumab and treatment interruption, and the restart criteria for treatment resumption were more than 4 weeks sustained viral load, more than 1,000 copies of more than 30% decline in CD4 T cell count from baseline or less than 350, obviously HIV or other clinical risk. symptoms, and pregnancy. Here you have the schedule of this clinical trial. As you can see, you have 9 vedolizumab infusion, 2 after treatment interruption, and 7 during the phase with RT treatment. You have an ATA phase of 26 weeks, and then a follow-up phase of 24 weeks. Unfortunately, there is no clear evidence in this clinical trial that vedolizumab may help to control viral load, and it was the conclusion of the authors. However, when you look more deeply in all the results, looking at all individual results and not the global impact of the treatment, you can see that around 1/3 of the patient reach weeks 26 of treatment interruption without meeting of criteria to restart ART. Importantly, it's also important to note that in this clinical trial, there was no placebo arm, and the comparison was made with an historical placebo control arm. We have also a second clinical trial, that has been performed with vedolizumab, made by Canadian team. You see here the inclusion criteria in this clinical trial, and here the aim was more. Once again, there was no placebo arm, and the aim of this clinical trial was to compare the impact of 2 different dosage of dose of vedolizumab, either 300 milligrams or 150 milligrams. You have only 7 injection of vedolizumab, and importantly, in contrast with the previous one, you have 4 injection after the treatment interruption. The conclusion of this clinical trial was that there is a clear dose effect of vedolizumab, and it seems that you may have some kind of control of the viral replication after treatment interruption with the 300 milligram dosage of vedolizumab. Therefore, if we want to summarize all this clinical trial and all the experiment performed in NHP, there is some kind of inconsistency in macaque studies that cannot be easily explained. We have 2 clinical trials, both with some signals of an impact on viral load in participants, and we have, as mentioned, no randomized controlled study to date. So therefore, it seems that you may have some effect on vedolizumab but needs to be clarified with a new clinical trial. So we have decided to build a new clinical trial with 2 new important points. The first one is to have a placebo arm in our clinical trials, and the second one was to combine vedolizumab plus MVA, therapeutic vaccine, because we have shown in a previous study performed here in France that we may induce CD4 and CD8 T cell responses with the MVA immunization in our patients. The aim was to look at the impact of the combination of MVA and vedolizumab on viral control, and we used the area under the curve, which seems to be the more interesting parameter to look at least if you look at the results of the Canadian study. There is a clear synergistic action between these 2 compounds, with a potential of the HIV-specific CD8 T cells with the MVA, and a reduction of the trafficking of CD4 T cells with the vedolizumab. So the objective is to evaluate the virological efficacy of both the combination of vedolizumab, either or not in combination with the therapeutic vaccine MVA, HIV-B in the HIV infected patients undergoing treatment interruption, and compare and looking at the control viral replication using area under the HIV RNA curve. Obviously, we look also at the safety of this strategy in the patients. So here you have the schedule of this clinical trial with 3 different arms, with combination with either the vaccine, MVA plus vedolizumab, or a placebo plus vedolizumab, or a placebo arm. And here you have the sample, the calculated sample size, which is currently 69 patients with 23 for each arm. As you can see here, we have kept the same kind of schedule that the Canadian one, meaning that we will have 3 vedolizumab infusion before treatment interruption, then 4 monthly injection of vedolizumab during the treatment interruption phase that will last at least last 24 weeks. So if we want to compare our current design with the other one in terms of number of vedo infusion, vedo dose, inclusion criteria, and when to restart ART, it seems that we are more close to the Canadian study with 7 vedolizumab infusion, a use of 300 milligrams. We will include patients with high number of CD4 T cell counts, both at baseline and with nadir more than 300 cells per millimeter cube, and obviously patients that are with undetectable viral load under treatment. The criteria to restart are listed here. Why did we choose this kind of criteria? Because we know with our previous clinical trial performed in France that there is no issue to reach even high number of copies, so higher viral replication at the peak, if we respect all the inclusion criteria at baseline. So just a few words now to end and to have obviously important knowledge on the impact of MVA and its role on the protection against the monkeypox viruses, because as you may be aware of, the vaccine used for to protect against monkeypox is an MVA vaccine. We use a different strain of MVA, but it's not so far different from the one used and recommended for the vaccination against monkeypox. So to try to summarize what are those type of vaccine, the pox viruses vaccine that have been developed initially historically against smallpox. You have the first generation of vaccine, namely the Dryvax vaccine, but which was associated with some kind of severe events. So there is the generation with time of different type of new vaccine. All these vaccines are live attenuated vaccine, but at the end you have the MVA vaccine, which is a very highly attenuated vaccine with has been cultured more than 570 times in cell culture, leading to an important deletion of its genome for more than 50%, meaning that you have a very safe vaccine at the end. This kind of vaccine have been used in the past in Germany to vaccinate against smallpox, and some new clinical trials and experiments in animal models show that it could be also efficacious against the monkeypox viruses. It has been demonstrated both in human And in human, you can induce with this vaccine antibody and T-cell response against both smallpox and monkeypox. And in non-human primate model, when you vaccinate the animals with the MVA, you completely protect them against the disease and against the death, because monkeypox is able to induce deaths in NHP animals. Just to end some results regarding the response against both smallpox and monkeypox in people living with HIV. And it's quite important to keep this in mind. The first one was an article, an experiment performed in people that have been vaccinated during childhood against smallpox. And the authors look at the persistence of immune response in subjects after they have been or not infected with HIV. And what you can see here that while it seems that there is maintenance indefinitely of antibodies in people, in HIV uninfected people, you have some decrease of the antibody response in people infected with HIV, associated with a clear decrease on the CD4 T-cell response against smallpox, while there is some kind of maintenance of CD8 T-cell response. So what are the conclusions of this kind of study? It means that even in older population, you know that in older population that have been vaccinated during childhood with a smallpox vaccine, there is no recommendation to vaccinate with MVA to protect against smallpox, or you can do only one shot of a vaccine. It could be quite different in HIV-infected people. What is now the immunogenicity of the MVA vaccine in people living with HIV? This slide could be quite complicated, but at the end, they look at both people that have been vaccinated in the past with against smallpox or not, and then they vaccinate or not them against monkeypox with the MVA. And at the end, it seems that the antibody response against with the MVA and against monkeypox, it's nearly the same in HIV-infected people than in HIV-uninfected people. Just one quick slide to show you that the MVA vaccine, the kind of MVA used in our clinical trials, have been used by other teams, and the use of this kind of vaccine are associated with a very good safety profile. So in conclusion, we want to look at the efficacy of the combination of VEDO and MVA to control viral load. And it's quite important for us because it will be, we have some elements to show that show us that it's quite relevant to use vedolizumab, but we don't have currently any kind of clinical trial with a placebo arm, which is quite important to make a definitive conclusion. And we want you to be involved in such a clinical trial to be to try to have a definitive conclusion. There is some potential aspect, impact of the MVA that will be used also on a monkeypox infection, but we don't know yet if we are allowed to say, for instance, that people that will receive our MVA we won't need, for instance, to be vaccinated against monkeypox in the next weeks, and we may discuss this at the end. Thanks a lot.
Thank you, Jean-Daniel. I can see a question from Axel in the chat box. Axel, do you want to unmute yourself and ask yourself your question, or should I read it?
It's like you wish.
Yep, go ahead.
Well, I saw just the results of the vedolizumab, and I'm wondering, because I don't know the mechanism, if you are also searching for, because this is typical, a more potent drug instead of combining with other drugs to create some synergism. Is there research ongoing, let's say, to find maybe a more potent drug to get more patients with a positive response?
If I understand your point, the question is, our goal here is not to try to develop new drugs, but to try to see if the available drugs could be interesting in terms of HIV cure, any perspective. And it seems that in this case, vedolizumab could be interesting. And it's for us, it's also interesting to combine them with the MVA vaccine. But our purpose is not to try to at this stage to try to develop new drugs, but only to look at the impact of current available drugs.
Okay.
If I may add on this, Jean-Daniel, if I understand well, in the Canadian study, there is no analysis of the participants' characteristics that could allow researchers to identify those where vedolizumab seemed to be more successful in controlling.
No. In the main difference in the Canadian, you have very few, a little number of patients, and the only conclusion was that it seems that the 300 milligrams was more powerful than 150 milligrams, and it's the only conclusion we can draw from this study.
Okay, thank you. So if there is no other burning clarification question and so on, I would go on with our agenda and ask Christel to present about the social science study, which is associated with the main study.
I think there is a last question just now.
Yes. Yeah, thank you, Christel. Jean-Daniel, last minute question. Is Vedo going to help with eliminating the virus from the head reservoir as well? Do we know this?
Sorry, I was on mute.
Yeah.
We don't know yet, but in the secondary objective, we will also look at these more directly on the HIV reservoir. However, if we have an impact on the area under cover, it means that we add an impact on the reservoir. So it's a different way to look at the reservoir, to look at this kind of parameter.
Thank you. Thank you for your question also. So, Christian, it's up to you. If you want to share your presentation and start it, you're very welcome. Thank you.
Okay, it's okay. You can see my screen.
Yes.
Okay, perfect. So it's very nice to present the social science part of the EHVA, which is about expectation, motivation, and experience of HIV patients who are going to participate to EHVA. And I will make a quite brief presentation just to introduce the general framework. So first, a little of context. So currently, antiretroviral treatment are well tolerated and cause fewer side effects, but do not cure the virus, mainly because of the viral persistence in cell reservoirs. So persons living with HIV have to take treatment at least once a day, every day for the rest of their lives, with now some possible treatment reliefs. And in addition to the difficulty of treatment adherence for such a long period, daily medicine is a constant reminder of the disease. And finally, risky behaviors and stigmatizations are still matters of concern. So in this context, HIV cure clinical trials raised hope, but hope that can be counterbalanced by ethical questions regarding trial participation. Indeed, they target controlled patients with normal life, if even there is a normal life. And participating in such trials may have consequences on daily lives because of treatment administrations and follow-up procedures. And moreover, there is no immediate direct clinical benefit except the possibility to benefit from an interruption of antiretroviral treatment with a close medical follow-up. And more specifically, treatment interruption could lead to increased viral load and increased HIV transmission risk, which with individual and collective consequences. So consequently, motivations and barriers have been extensively questioned during the 5 last years, and showing the importance to explore and to document participants' and decliners' expectations and experience in real life to better inform and to better support participants and future participants, and to help improving the design of future HIV cure clinical trials. So just some results we have obtained from hypothetical situations will give us some information of the factor associated with the possibility to refuse to participate in HIV cure trials. So regarding the person living with HIV, the odds to refuse to participate will increase for the ones with a precarious financial situation. The odds will increase also with the ones who have the highest educational level and the lowest educational level, which is nearly the same result than for the vaccination in general. And it will also, so the odds to refuse to participate increase through for the people who thought that current antiretroviral treatment would not continue to be effective in the long term. On the reverse, the odds to refuse to participate decrease for the persons that perceive themselves as belonging to the person living with HIV community, and for the ones who are very concerned by the risk of transmitting HIV. There is some information also regarding the physicians and the odds to refuse to propose some HIV cure clinical trials. So the odds to refuse increase when the physicians participated in the most recent clinical trial because the department head asked, and when they believe their patients were concerned about HIV negative impact on their professional lives. On the reverse, the odds to refuse to propose participation to their patients will decrease when they are favorable toward participation in clinical trials in general, which is obvious. For the physicians who are more research-oriented, and when they participated in actions organized by HIV associations, and when they inform themselves about HIV cure research with international scientific literature, but also with HIV associations journals. So the objective of the social science part is to explore and to document. We have 4 specific objectives. So to explore the evolution over time of expectations and motivations related to the participation, the evolution of the experience of this participation, and the satisfaction with the information delivered. We are also interested in anticipation and understanding of risks and benefits related to participation. And there is a focus on the experience just before and during the analytical treatment interruption period. So we're going to look to the anticipation and the representations of the ATI, ATI in English, and the impact on preventive sexual behaviors and sexual quality of life. And we are also interested by the motivations and the experience related to refusal of participation. So to reach this objective, we use two components, a quantitative one in all the participating centers. So there is 4 self-administered questionnaires that were first modified after a discussion we had. There is a few years now in Brussels, and we have had a COVID-19 module, and it will be proposed to each trial participant in France, UK, Switzerland, mainly. And there is a qualitative component only in France, where we are going to use semi-directive individual interviews with trial participants and with trial decliners. And I don't know if we have to think to a very brief anonymous questionnaire for all the centers in EHVA who participated in EHVA. And if any this will happen, we also will conduct some individual interviews with the individual, not include because of a negative screening result to collect their experience. So it's a voluntary participation. There is a guarantee of data confidentiality, and there is a specific consent form for this part. So just to give you an overview of the 4 questionnaires, as you can see, there is some module we just asked for the inclusion, and some of them were repeated to have longitudinal analysis. So we are going to explore sociodemographic characteristics, experience with HIV and daily treatments, expectations, motivations, and fears, understanding of the risks and benefits, satisfaction with information and with feedback, which has been shown to be very important, and trial consequences and symptoms perception, quality of life and mood, and sexual life in terms of quality and preventive behaviors, and after that, the retrospective perception of the trial participation. Regarding the individual interviews, of course, we're going to focus on motivations to participate, expectations, general experience, and with a very focused on the HEI period. And for the decliners, we are going to ask them the reasons to decline, the reasons that might have led them to participate, and expectation and position regarding the future therapeutic vaccination trials. And of course, individual interviews are totally anonymous. So just to give you a very brief overview of this part, because I think we have already discussed it. Thank you for your attention.
Thank you, Christel. Thank you very much. Is there any question related to the presentation of Christel of this social science study? As you can understand, we don't have so many cure studies running in Europe, so I think that having a tool to assess also the satisfaction, the motivations of participants is really, really important. No question? Yeah, we have a question from Robert. Maybe I might have missed the info. How many people are participating to the study? 69. I wrote it in the chat box. 69 participants total for the main study.
So for that point, I think Jean-Daniel or Sheena can better answer than me.
Yeah.
You mean for the social science? I mean, we're hoping that everybody will consent in France and UK and Switzerland. Christel, I think we didn't translate it into German in the end.
No, because when we changed from EHVA T01 to EHVA T02, the expected number
This is very small.
in German was very, 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 very low. So
yeah, no,
we are not included for this reason.
But I think it's a very interesting question that Robert raises, Giulio, about Jean-Daniel, how many we will be able to recruit. So we're up against some challenges with the timeline and things, and that is a challenge. And I have that, I think I did try to raise that in my presentation. So perhaps we could, we could.
I think it's the right time to do so.
Yeah.
Shall I carry on?
No, but please, we'll see. Yes, go ahead.
Okay, I'm going to stop my camera in case of internet and just for the presentation and come back to it later. So it is, as everyone else has said, a real pleasure to be here with you. I'm terribly grateful to you for organizing this, Giulio, Giorgio, and the EATG team. I hope that is in presentation mode. I think it is. So what I wanted to do was just quickly remind people, and I know many of you know this already, but the development of the informed consent with the community and trying to identify what the key elements were for understanding. And then we've had to adapt to events which include bankruptcy, COVID, and monkeypox to a lesser extent, really, but it seems life is ever changing. And there are other challenges, practical, feasible challenges ahead, and we are thinking about how we might need to adapt again to address those. So the grant actually started in 2016, and the grant application at that point in time, the design was fleshed out in the grant application. We started monthly trial management group calls towards the end of that year. And between 2016 and 2019, we were working on T01. And T01 involved two different vaccines. We didn't have vedolizumab, and we had actually enrolled one participant who luckily was enrolled into the group that didn't receive any treatment for 2 weeks, and the product provider went insolvent, and we had to close that trial immediately. In 2019, then we redesigned T02. And the community have been represented not only on the TMG, which Giorgio and Giulio have attended. We also have Simon Collins on the Trial Steering Committee and David Herry on the Independent Data Monitoring Committee. So definitely the TMG has the most input into development, but the Trial Steering Committee also has a big say, and then probably less so the Independent Data Monitoring Committee, because they're a step back, being completely independent. In 2017, with Giulio and the group from EATG, and it was Mitzi who drove this with you, Giulio, we developed a participant and public involvement strategy. So then we have very much, EATG have been completely involved in the protocol development through their participation in TMG, but really driven the development of the information sheet. Very, very much so, and circulated more broadly within the network. So really key to ensure that participant expectations of the interventions were realistic. We'd never expected them and do not expect them now to be as effective as combination treatment. So there are, of course, inevitably risks for the person, as well as the risks of onward transmission in the treatment interruption period. And the access to PrEP through the period in which we've been developing this has been patchy, was really only available in France, but now that has changed and it is available in all participating countries. some aspects needed more thorough scrutiny. And it's lovely to have Richard on the webinar today. And we're very grateful, Richard, for your input into the language that we were using. Richard, I feel you have more expertise in treatment interruption trials than I do, and it was great to have your review. And then I've had a couple of opportunities to sit down with Simon as well, so that's been very helpful. And then further changes that we had to make. I don't know if I maybe can't get rid of that bar at the top. I do that? No, I don't think it will. Further changes that we've had to make, of course, have followed the review by the ethics committees, who were also completely independent of the trial team in the 4 countries that had reviewed it. So that 4 countries did include Spain at the time. And then EATG organised this community meeting that Christel mentioned in November 2018, and many members there expressed their concern about the language used in the information sheet. It was very long, it was complex, and unfortunately, you know, we're a bit tied in with what we have to do according to the regulations and the guidance. So some things we cannot change, but we were able to take forward the suggestion of a summary leaflet, a briefer summary leaflet, so that the first glance at the trial was not completely overwhelming. I mentioned that TO1 started in CHU in Switzerland in February 2019, and we're very grateful to CHU, who've been our lead centre throughout EHVA. But as I said, because of this bankruptcy of one of the vaccine providers, we had to stop that trial 2 weeks after enrolling the first participant. And redesigning the trial actually took a while because we decided to incorporate that monoclonal antibody vedolizumab, and we had to sort out access to that. That was a commercial product. We had to work out how we could access it. We had to recalculate the sample size. But to be honest, the key elements in terms of the risk of the treatment interruption didn't really change to those we'd already discussed in developing TO1. So the intensity of the schedule, the onward transmission, the personal risks of stopping treatment all really remained the same. But our STAR Centre started her screening again in February 2020, and we all know what came next, the pandemic. So over the course of that year, everything was on pause. We were reviewing the eligibility with respect to the risk of more severe COVID. And notably, our upper age limit goes up to 65. We had a nadir CD4 threshold of 300. So you remember the Canadian study allowed 200. We were allowing 300. We also allowed people who had just Kaposi sarcoma in as well. It was unclear at the beginning of the pandemic what the impact of COVID was going to be on people living with HIV. Would they have a greater risk of severe COVID? But even if it wasn't the case during when they were on combined treatment, perhaps this would be likely to be the case in the treatment interruption. So that was obviously a big concern. We didn't actually have, although it took a while to get this information, but we didn't have concerns about the actual interventions and COVID having an interaction. And then COVID research also took all of us over, you know, facilities, resources, everything. Non-COVID research was really paused globally. So then we had a meeting again. Giulio organised this, I think, or maybe this was organised by IAS actually. Yes, coordinated by IAS. This was also in November 2019. Is that right, or was that 2020? I'm almost losing sight of the years hit now. And Roger Tattoo presented a proposal towards a consensus statement on treatment interruption trials in the COVID era. Jean-Daniel presented the EHVA T02 proposal and how the risks would be mitigated. And Giulio, you led the discussion. There was certainly a concern about restricting other vaccines in the study. At the time, we didn't have COVID vaccines, but we did have flu, and we didn't want for people to not have access to flu vaccines. And travel restrictions were intermittently in place due to lockdown. So we knew that the weekly visits for viral load would be really, really challenging. But there were treatment interruption trials that were ongoing in Spain and on the west coast of America, that people had managed to keep going in spite of the pandemic. So we felt, you know, overall, we acknowledged that healthcare and non-COVID research did need to resume at some time, and we had to find a way to do this with minimal risks. But a month after our meeting, the authorized vaccines for COVID were rolling out in Europe, and that completely changed the complexion of the protocol. So the eligibility was revised, must have completed an authorized vaccine regimen, 28 days or more prior to enrolment. We raised the CD4 to 350 and we excluded Corbelli. And then of course, treatment would restart in the interruption if there was lab confirmed COVID. So we added that as another criteria. We clarified that during follow-up, there should be a 28-day gap between study injections and authorized vaccines, if possible. That should be maintained. And 2 centres are open to recruitment now. So CHU screened the first participant on the 10th of May and enrolled on the 21st of June. And then Chelsea and Westminster have their first screening booked for the 19th of July. And I think the second one's booked for the 10th of August. And Henri Mondor, Jean Daniel's centre is anticipated to open in the next month, and the 2 other centres in Paris to follow. And then the centre in Hamburg is responding at the moment to ethics requests, so that's a little bit further on the line, but they do have regulatory approval. So we're hoping to open in the autumn. But there's a couple of things we really would like to raise in terms of where we are now. I think we've all seen COVID cases rising everywhere in all of our countries. We have to consider, you know, the possibility of reduced COVID vaccine efficacy with new variants. So I think it's very important that the ATI stops in participants if they have confirmed COVID. It's also important to know, for participants always to know, that they can choose to restart at any time, as can their clinician, if that's what choose to recommend that, if they are concerned. So we believe that even if that does evolve, that situation over the next winter or the next spring, that these mitigations we have put in the protocol will still be good enough for that situation. But we'd be very interested to hear your views on what the community feeling about the threat from COVID is, and whether this plan is acceptable. So that's one issue. And then the other thing is the challenges that we've got. You may have noticed, I'm not sure if those of you knew, but definitely Giulio and Giulio will know that at the beginning of the grant, Amsterdam was going to take part, and we lost them quite early on. But IDIBAPS in Barcelona was a major partner, and because they've done a lot of trials, both cohort trials and cure studies, we were expecting quite a large number from Barcelona, and then a smaller number from INME in Rome. And unfortunately, you know, we've lost these 3 centres, but funds, you know, can be redistributed. Larger numbers are needed from the 4 centres that are participating. Lausanne and Hamburg have limited numbers living in those cities, so that will be a limitation. But the populations in London and Paris are really substantial. However, we have to acknowledge that travel in those cities is time-consuming and can be very expensive. The duration of follow-up, if a participant completes the 24 weeks of ATI, can be 54 weeks. We have to allow for this plus the 12 weeks to close out centres. So it can be 66 weeks really from the last person being enrolled that we have to think of managing. And clearly in the current grant end, we can't do that because it's June 23 already extended. So we need to request a further extension. And even with a further extension, I have to say, because we only have the 4 cities taking place, achieving that sample of 69 by the end of Q1 2023 still looks pretty challenging. So Robert, your question is very, very relevant. Can we reach the 69? We've allowed for a loss to follow up of 10% because of COVID. But it may be more than that in the end. So this would mean ending up with an underpowered trial according to our assumptions. So I thought it important to go back and discuss with you what the assumptions were. So we're working with an endpoint that's the area under the HIV RNA curve in that 24 weeks of ATI. So the 24 weeks of ATI is what all of the calculations are based on. Although it's chosen as an overall summary and chosen particularly because vedolizumab works when virus is replicating. So if we were to use the time to rebound, we would underestimate effectiveness. 16% based on the Dahlia trial are anticipated to recommence before the 24 weeks because they hit that viral load endpoint, resulting in an apparent lower area under the curve, which is just the way it works. But the team in Bordeaux can actually model what would have happened using the Dahlia data, and they can accommodate for that. As I mentioned, 10% may not start the treatment interruption or may lose them for other reasons. We've got a two-sided significance because there are two active arms to compare to placebo, and 80% power with 69 people enrolled is where we are. So I think I want to come back to the other points about EHVA T02. It's the first randomized placebo-controlled trial of Vido. There will be 20 individuals who are exposed to Vido in our trial, but in contrast to the first trial where they compared to historical controls, we will have a randomized placebo control. And the first trial only gave 2 vedolizumab in the ATI phase, whereas we're going to give 4 in the ATI phase, as the Canadian study did. The Canadian study only did a comparison of the dose. That's a typo, sorry, 150 compared to 300. And so it was small numbers. And really, as Jean-Daniel said, the only conclusion we can draw is about the dose. There have been a large number of therapeutic vaccine trials with limited success in terms of control in viral load, but none of these have combined a vaccine with monoclonal antibodies, and synergy of these two products is plausible. So I think the trial management group felt that it would be perfectly justifiable to describe the trial we're doing as exploratory. We have put in, a sensible sample size in the protocol. We have talked about 0.6 log copies being the equivalent of that, the area under the curve being a significant difference between the groups. And we may yet observe that even with a sample of only 30 participants. If we do observe it with 30 participants, it could not be significant because it's underpowered. If we don't observe it, then we may not be able to exclude the possibility that the difference was that good because it was underpowered. So those are the compromises that we'd be making with a smaller sample size, but we would still have a result of this monoclonal antibody compared to a randomized parallel placebo group. So I think I'm right in reflecting the views of the trial management group that we felt comfortable with this if we managed to achieve 30 who managed to enter the treatment interruption, then we're going to produce a valuable result overall. And when we ask the centres the numbers that they can recruit, 30 feels feasible over this course of time, which would extend itself into 2023. 69, I think, does not feel achievable. The things we considered were changing the primary objective to focus on Vido. In this case, we'd have 80% power with a sample of 40, but we would have to increase the chance of being allocated to placebo, and we would require a protocol approval. So to be honest, it's not practical in the timeline for us to pursue that. And ultimately, our chief investigator was not in favour of this proposal. The other thing that's been mentioned was to change the trial to reduce the ATI or to remove the ATI. So if we were to reduce it, our power estimations would be invalidated. If we remove it, it really becomes a different study altogether, and we couldn't do it under this trial protocol or this number. So as well as wanting to know what you feel about the threat of COVID, the final questions really are whether you feel that we're embarking on a trial that's worthwhile, even if we only manage to enroll 30 participants who start the ATI. Should we go back and consider refocusing on vedolizumab? And finally, you know, of interest really, because this definitely wouldn't be practical in the timeline, what are your thoughts about a study of these products with no ATI? Do you think we could actually assess them if we were to remove it altogether? So Giulio, these are big questions. And I don't know if you'd like me to leave the slides up or if everybody can remember them.
I think it is good to have this slide for
Yes.
leading the discussion. Yeah. So we have a very first comment from Sergio in the chat. If Sergio, you can unmute yourself and speak in the group if you wish, or I can read your question. If we remove the ATI, how would the efficacy results be measured?
Jean-Daniel, do you want to answer that? I mean, I can't see how at all myself. I think the total DNA would give, you know, you might have little blips in that that are different.
Yes, obviously, if we remove the ATI, we need to use a different endpoints, and either DNA or other biological endpoints of the same kind of biological endpoints. However, that are less relevant, obviously, than the area under the curve, but it had been used by other for HIV cure without ATI. They are not perfect, but they can work in some way.
Thank you, Jean-Daniel. I would like to listen to the opinion of other participants to this webinar related to the questions that Sheena raised.
This is Richard. I think these are difficult questions. I really appreciate the transparency around the sort of the challenges or all the practical challenges involved in trying to do a study like this. We don't often have a window into all that kind of stuff. I mean, to me, I think because there are still lingering questions about the antibody, I think it probably would still be worthwhile to do if you can only enroll 30 people, because at least there's a placebo control, and then it seems like a reasonable chance of getting more clarity around that.
That's what we suppose, Sheena or Jean-Daniel.
Yes, it is exactly. I mean, it's why we've kind of talked about 30, Richard, on the TMG, because that would then be the same number that have been in the largest clinical trial to date, you know, exposed to Vido, taking advantage of what we learned in the Canadian study. And so if there was that level of difference, you know, that equates to 0.6 log, you would have an observed estimate that hit what you were saying was your estimate of interest. So even if it ended up not being significant because the number was too small, you would have shown a result in this trial, which I find difficult to say is anything other than exploratory, because it is the first time these products have been combined together, and the first time you've had a valid comparator. So whilst Vido has been looked at before, MVA vaccines have been looked at before, this, in my mind, retains an exploratory nature because it's the first time you put the two together.
Again, a question from Sergio. Can the initial target number of participants be raised from 69 to, let's say, 100? well, I think that the problem.
Oh, oh Sergio, no. I think I think we're feeling 69 is quite hard, but you really can't tell until you start, and trials can gather momentum as people speak to each other, and so that's certainly possible. And I think the summary leaflet that you helped us to develop, we'll be able to distribute that in clinics, and you just don't know who will pick it up, and people might be interested. I was really interested in Christel's presentation, and Christel, did I understand this correctly, that that interrupting treatment is part of the motivation for taking part in these trials? I wasn't sure I got the arrow right there. Is Christel still with us? Yeah.
Yes, you have well understood. Yes, it's one of the motivation to participate in HIV cure. It's both a motivation and a fear. Could be the same. Could be both of them. But yes, it is the motivation to benefit of an interruption with a close monitoring.
And I think for me that's the main reason that this last bullet here, to change the protocol in the middle when we have enrolled people who believe they're taking part in an ATI trial is not, is not right. I think that's that would not be right, even if honestly, to be fair, it can't be done. As Jean-Daniel says, it completely changes the study. It would have to be a different protocol. So I think the number we can enroll, Sergio, is unknown. 69 feels like a challenge, but 30, is doable based on what centers have told us at the moment. So I think CHU feel they could probably enroll 10 participants in Lausanne, and Jean-Daniel across the 3 centers in Paris, they can enroll 15, and in Chelsea 10. But if we're able to get those samples quicker in the larger cities of Paris and London, then I don't see why we can't get beyond that. To get double that feels a stretch, I must admit, but I do think we will be able to get a number that will provide us a very valuable insight on the performance of this monoclonal antibody, and allow us, I think, to understand a little bit more about the mechanism regarding how it works. That will inevitably take longer to iron out, because it will depend on very detailed immunological assays, and they always come quite some time after the trial.
The recruitment obviously is the key point. For instance, we have here in Mondor, we follow a cohort of 1,300 participants. If you look at only the people who with the good criteria of inclusion, you have around 50 people, and at the end only 3 to 5 want to participate in the clinical trial. So, and we are not sure that they will participate at the end. So we need a very large cohort of patients to be sure that we will able to include even 30 participants.
Sorry, Jean-Daniel, I could not understand. This evaluation of the availability is based on a questionnaire which was administered.
No, we have the large cohort, so we have the inclusion criteria. So we screen our cohort with the, and we add at the end around 50 persons. So we discuss with them, we exchange with them, and try to see if they want to participate. And at the end, we only have a positive answer. We have, we have always inclusion criteria that are not in our database. For instance, history of cancer that are not written in the file, and so on. But at the end, you have 1,030, 50, sorry, and at the end 3 to 5.
Okay.
Meaning that to have 3 patients, you need to, for 1 patient, you need to have in your cohort 500 patients for.
Wow. Which is a lot. I anticipate this is the same situation happens in the other sites, at the other sites.
I think that's true, but because Marta was, I don't know if I can get these numbers right, but I think Marta was saying they had gone through, you know, the file data for 300 people living with HIV for another cure study in order to identify 2, and one of them had not in the end decided not to enroll in the end. But they were
sorry, Sheena, a little bit more.
You're there. Okay.
Sorry, thank you. Yeah, no, but you're right. So we had a list for another cure study of 300 plus, and about 6 or 7 were eligible and expressed an interest. So that's our experience.
But I think we've got slightly more generous criteria in EHVA T02.
Absolutely, there was a study. Yeah, I agree with you, with much stricter criteria. Let's say, if you just want to kind of very high level, that would be 50% or less of people eligible for that study would be eligible for a, sorry, 100% would be eligible for EHVA, 50% would be eligible for the other study. So we would have 1,000 people to be able to screen at least, or more.
I was asking myself how to put together this situation with the big expectations that I feel in the HIV community towards the cure research. I mean, within my community, there are so many people who are very anxious to know more about HIV cure research, and many of them are willing to contribute. So beyond the eligibility criteria, I know that there is a willing to participate, a willing to contribute. At least this is my perception. Unfortunately, we do not have in Europe such an extensive social science research and HIV cure research that can inform this kind of considerations. But yeah.
Can I add something, Giulio?
Sure.
Before COVID, yes, I agree fully. I think the whole population of humans have been overwhelmed during the COVID pandemic, because we noticed that a lot of people just say, no, thanks, I can't do it. There was this much more exhaustion related to life in general. So I don't want to sound negative, but I would do 100% with you. Before COVID, there was so much enthusiasm and there were happy conversations about everything. At the moment,
if I may, Marta, I disagree in a sense with you. I don't know, especially for HIV cure, but for instance, for HIV prophylactic vaccine and for the healthy volunteers, we did not face any kind of issue to recruit in our last HIV prophylactic vaccine, and it was easier than before the COVID era. So,
Daniela, I agree with the positive people who are not living with HIV. You know, you always find them, but I don't know. We are struggling. I think we are struggling a bit. They just, yeah.
Yeah. It's pure speculation. So there is also another question from Sergio in the chat box. I don't know whether you read it. How about continuing the study with those who stopped the ATI period as well, and analyzing them with other tools than those who continue with the ATI? I'm not sure I understand that question, Sergio.
I think I understand it, but Sergio, do you want to say you mean people who interrupt the treatment interruption early, who go back onto treatment before the 24 weeks is up? Yes. So that that is where the team in Bordeaux have really helped, because they are able to use the data from previous treatment interruption trials to model what would have happened going forward. So to predict the whole area under the curve based on what they have up to whatever point it is that either the participant decides, the clinician decides. I think we can't do it for people who never start the ATI, but for people who start the ATI, we are able to use a mixed model to offset that. Is that the correct, does that answer your question, Sergio?
Apparently, yes, because you wrote great.
Oh, is it great? Okay, lovely. Yeah, yeah, great. Great.
And we have another question from Axel. Is there a I don't know whether you want to ask your question yourself, Axel, or I can read it.
No, go please.
Okay. Is there a sub-study possible with the persons who respond positively, so that more arguments could be used and be more selective in the criteria?
No.
Are you thinking of Christel's study here, Axel?
No, we need a lot of people to predict what will be the one who will answer to a positive response to the treatment. So it's not possible to currently to perform such kind of study. You mean you include some people, you look what are the parameters associated with the good response, and then you change the criteria to include more? I am correct?
Yes, that I was thinking about.
Yes.
Because in my point of view, you need to recruit so many persons that it's very, very difficult to achieve in reality.
Yes, but to try to answer to your point, you need more people because you need to decipher what are the parameters associated with the good response, which could be quite difficult.
But at this stage, you cannot do it.
No.
Okay.
I think it's quite complicated, Axel, also to disentangle it from the responses, you know, that are natural to an individual that help with the control of virus. And they've been richly studied, and yet I don't know that we still have we have, you know, clarity about what looks helpful, but I don't know if we have real confidence in all of those.
Okay.
This might be confidential, but I was just curious, was there a reason why the Barcelona site was that funding related, that why they didn't continue?
No, it was what can we say? There were administrative difficulties that made it impossible for the PI to continue. Yeah, and we lost the PI really. And we're deeply saddened by that.
Sure thing. No, I understand.
And then for Italy, we ran up against a regulatory obstacle. So although, you know, we had presented it as a phase 2, the Italian regulatory authority, although, you know, they looked on it as a first-in-man study because it was the first time the two products had been combined. And therefore they were asking for an awful lot of detail that we weren't in a position to provide. And so in the end, that was impossible. And I think the Italian team in particularly have really been distracted by the COVID pandemic, followed by, you know, trying to restart everything else, you know, health service and all the rest of it. But that was really a kind of regulatory obstacle we ran into there. And then Amsterdam, I can't remember, I sung on the call still, I've actually forgotten now why Amsterdam, but they didn't want to take part, I think they changed their mind.
Yeah, correct. No, they actually they were never in.
It's
a long history, but they never won from the beginning. They were never part of the trial.
Yeah, they didn't want to take part in EHVA T01, did they? I think we'd lost them already at the beginning. Yeah. I don't know, Richard, while we have you, do you have an update on the global situation with ATI trials? Do you know what's happening worldwide at the moment? What's new in the pipe?
I mean, I can only work from what's in the registry entries, really. So I'm not in like sort of contact with people very much. I think there's around 30 ongoing trials that have ATIs in our most recent listing. So, and there were a couple that paused due to COVID that have restarted. And I think right now there's only it's not an ATI study, but I can only think of one study that's still paused due to COVID. So most of the research does seem to be getting back underway.
Well, that's good news. I see Robert's put a question in there and asking about the disadvantages if we were to refocus on vedolizumab alone, given that the number of participants is very small for comparison between those who have VD on its own and those who have it in combination. And Robert, you're quite right, even with 69, we didn't have the power to compare the two active groups. It's always possible you will observe a significant difference just by chance, but not by chance, but by good luck, if you like. But we didn't, we didn't have the power to do that. And we really, really didn't have a way of, well, we said that 0.6 log would, the equivalent of that with area under the curve would be a clinically relevant difference. And we didn't anticipate that the synergy between the two products would be that large. So I think we don't have the power to do that comparison even with 69. So I think the real disadvantage is refocusing on vedolizumab is twofold. One is we would have to alter the protocol. So we would enroll at the moment, 1 to 1 to 1, and then we would have to change the randomization ratio to 1 to 1 to 2, so the chance of getting placebo would go up. Instead of it being 1 in 3, it would be 1 in 2. So we would need to change the information sheet for people we were newly enrolling. And we would only be able to do that when we had approval from ethics and regulators. So we might already have enrolled a significant number by the time we could make that change. So it probably wouldn't be 40 in total, but would end up being less than 69. But there are practical challenges, I think, in all of that to actually implement that. But most of all, for the community, it's the disadvantage of increasing the proportion, increasing the chance of being on placebo. I don't know what you think about that. Thank you, Richard. I don't know, Robert, if you wanted to say whether that would put people off.
Your answer clarified.
Okay.
Yeah.
I think the practicalities would get in our way. In many ways, we've been about to start this trial for such a long time that it sort of the time we first considered should we just not start at all was back in February, when we were looking at the end of the grant in June 23 and thinking we can't enroll everybody by the end of May, we just can't do it. But we had the sense that the commission would be sympathetic to a no-cost extension and that we could manage to eat the funds out long enough to make the enrollment go further. So we have gone on, and now the pressure is with the centres, with the community, to see what we can achieve. But I am an optimist. I think Jean-Daniel and Marta are optimists. I think Giulio, Giorgio, we are optimistic on the TMG overall. We
need to stay optimistic. However, clearly we need an help from you to communicate with the community, and I don't know what happened in other European countries, but at least in France, we are faces important difficulties in hospitals with we don't have any sufficient number of nurses to take care of patients. So I try to stimulate, to discuss with my colleagues, but there are so many other things to take care of, including the care of hospitalized patients, the new wave of COVID, for instance, right now to deal with the MBA vaccination for monkeypox. So it's very very hard to bring them with us on this clinical trial, but it could be quite different if we may have an impact directly with people living with HIV through your help.
Yeah. If I may leave my hat as a moderator and just explain my personal opinion about this, I think that redesigning the trial is something I would not be in favor of, because what Sheena just explained, because there is a huge delay, or it would be it will create logistical difficulties to conduct the trial and so on. And we already screened some people also, so it is a bit difficult in this situation. Anyway, so I think that, but in this view, the best option would be to try and do our best to enroll as many participants as we can. But the question is, do we risk to have a completely useless trial? So to ask people to join and to make such a great effort, because participation is quite complicated in this trial, for nothing, for something. I am optimistic in this sense. I think that we will be able to gain some important data, even though not conclusive probably, but this is not the trial that will give a definitive answer about the cure research. Of course, it is not intended this way, but we can have some very good information by this trial. And I think we may be able to enroll more than 30 participants. I don't know whether we will be able to enroll 69 participants, but I think 30 is a feasible target as far as I can feel. I also know the situation of prophylactic vaccine because I'm working in other studies, so I know how easy it was to enroll in this kind of trial, during the COVID-19 pandemic, during the lockdowns and so on. So I'm optimistic. I don't know whether among the other community representatives in the webinar, this optimism is shared or not. I would like to know so. Also, we are reaching the end of the meeting, so I would be happy to listen for final consideration from the community.
I guess maybe just one last point is just, I can't remember, but maybe to ensure that superinfection is mentioned as a risk for ATI, because that just happened in one of the NIH studies where somebody got superinfected when they went off.
Consent form, Richard?
I'm sorry?
Do you mean in the consent form?
Yeah, I mean, I don't know, it might be too late actually if it's but in the counseling, just because I know it's kind of usually framed as concerned about risk to partners, but there may also be a superinfection risk to the individual as well.
We mainly focused on the risk of HIV transmission, that's superinfection, but we may look, we may discuss this. Sheena, I don't know what is your feeling.
Well, I was going to have a look and see if we did. I don't know if anybody can. Would we have called it superinfection? Richard, I don't remember. It doesn't ring a bell. But I think, Giulio, we sent, I sent the global PIS and summary leaflet. So feel free, please, to circulate that to everyone. Yeah, he's on everybody. That's not a problem.
Okay.
Anyway, I think I can't find the answer to that quickly, Richard. I'm not sure we did.
I don't think so, but as far as I can remember, I don't.
No, I do. Probably I can't remember, but I don't think me and Simon would have probably raised it back then either. It's just something that came up recently, so something to bear in mind for the future, maybe.
Okay.
It happened in one of the Tai-Wook-Chan studies at NIH, where somebody developed a superinfection.
Anyway, since
we've certainly talked about the possibility that treatment would have to change. I think I should have mentioned that because that was another key element. That was another key element in the understanding that people might not be able to restart the treatment they had before if there was resistance. Yeah. But thank you for raising that.
Yeah, thank you, Richard. Anyway, since we are reaching the 90 minutes limit, if there is any final consideration or comment, I'm very happy to welcome it. Otherwise, please feel free to send an email to me, to Giorgio, to EATG in general, or to Sheena or whoever, if you have any other question or comment or suggestion. That would be very, very helpful. Okay. So should we close now? Thank you, everybody. Thank you, Sheena. Thank you, Christel. Thank you, Jean-Daniel. Thank you, everybody, for participating.
Thank you. Thank you for organizing.
And yes, please send an email for any question or comment you might have. And thank you again, and have a good evening. Bye-bye.
Bye-bye. Bye-bye.
Thank you. Bye-bye.